The overall goal of this procedure is to visualize chromosome-bound proteins by fluorescence microscopy. This is accomplished by first removing the cell wall by treatment with the enzyme zymolyase. The second step is to lyse the cell and nuclear membranes by osmotic shock, usually with the help of a detergent. This is done in the presence of a low concentration of fixative to preserve protein structure. After the cells lyse, the concentration of fixative is increased to further stabilize protein structures. The lysed cells are then deposited on a microscope slide and nuclei allowed to settle onto the slide surface. The final step is to immunostain the samples. The indirect immunostaining method demonstrated here uses a primary antibody that binds the protein of interest and a secondary fluorescently labeled antibody that binds the primary antibody. Ultimately, immunofluorescence microscopy is used to examine the distribution of chromosome-bound proteins. These representative images are of a spread meiotic nucleus immunostained for the recombination proteins DMC1 and RAD51.
The main advantage of this technique over the other commonly used method, which was introduced by Mike Dresser and Craig Giroux, is that most of the surface of the slide will contain optimally spread nuclei, making it easy to obtain images of large numbers of spread nuclei from one slide. This method can help answer key questions in the study of subnuclear structures, such as, do two proteins co-localize? Or does the formation of a particular nuclear substructure depend on the function of a given gene of interest? We emphasize that this method was originally developed by Franz Klein and Josef Loidl. We hope that this video will help others master our version of the technique.